Hi guys, here is the video voiceover for lab, I believe this is lab number 12. I'm losing track of my lab numbers, but this is a lab on exoenzymes. It's basically lab 510 through 511, 510 through 513. And as I'm talking right now, I realize I've not updated the questions that you should be looking at in these labs. I'll do that right after the voiceover. So fortunately, even though there's 4 labs associated with this lecture, they're basically all the same lab. And so this is a pretty straightforward lab. And the quiz and kind of understanding these concepts is really, is probably the easiest of the biochemical tests. So when we think about the term exoenzymes, let me grab, there we go. When we think about the term exoenzymes, remember exo means out, and enzyme, of course, is a protein that serves some kind of enzymatic function, and we're still kind of going along with this theme of nutrient utilization because all of these biochemical tests really are about a bacteria's ability to metabolize something specific and how it metabolizes that, whether it takes a fermentation route or anaerobic, aerobic respiration route, and so on. So the more tools that bacteria have and the more diverse those tools are, the more likely they're to survive in diverse environments. And then also think about this seems obvious, but bacteria can't eat in the way that we think. Like they don't have a digestive system. They can't just import things into their stomach like we can. And they can't perform endocytosis or phagocytosis like other larger eukaryotic cells can do. So how do they take in nutrients, right? That's the question. Like how do things from the outside get inside the bacteria? Especially given the fact that bacteria have a very complex cell membrane, cell wall apparatus that protects them from that external environment. So the only way that they can do this really is to import individual molecules from the external environment into the cell through either like active transport, a facilitated diffusion, sometimes simple diffusion. In the case of really like glucose, I think can get into the cell. Does it get in through diffusion? I feel like it's facilitated diffusion, but the idea here is that these molecules have to be pretty small in order to go through a specific kind of gated pore or get through the cell wall and cell membrane somehow. So all that to say that they can't take in these really large molecules that will provide lots of energy for them. So if they have the ability to, it would be really nice to take these large molecules and turn them into something smaller so that they can bring it into the cell. So let's remind ourselves a little bit about biochemistry. Remember that there's 4 major biochemical molecules that make up life and that living things need: proteins, nucleic acids, lipids, and carbs. And each of these more or less have a subunit that makes them up. Proteins are made up of amino acids that are stitched together. Nucleic acids are made up of nucleotides. Carbohydrates are made up, even really complex carbohydrates like starches are just long strings of sugar, simple sugar molecules like glucose or maltose or what's another, galactose, and so on. Lipids are a bit tricky because lipids are such a diverse group of macromolecules. Remember that a lipid is basically any molecule that's not a protein, a nucleic acid, or a carb and is hydrophobic. So lipids have a very diverse, they're kind of a diverse group. So we don't really have a straightforward subunit or monomer for lipids, but we can break them down into smaller pieces. So the idea here again is that bacteria need all of these molecules, but they can't take the larger macromolecule or the polymer into the cell. They have to make them into monomers or something, at least something smaller. So the idea here is that we have these exoenzymes that the bacteria can secrete out into the environment. They can take that big molecule that the bacteria can't import, chop it up into those smaller monomers or smaller subunits, and now we have a smaller molecule that the bacteria can in fact bring into the cell. So once it's in the cell, of course, it goes into the central metabolic pathway, right? If it's a sugar, it gets turned into glucose and then starts into glycolysis. If it's a protein, it'll probably undergo deamination or decarboxylation and then go into the Krebs cycle somewhere and so on. So let's look at our first, so we're basically going to do a test. Our our four labs this week our four labs this week are testing each of these four major macromolecules. So our first one is going to look at carbs. Remember that a starch is a type of carb, and if you look at this lab in the book, they have a diagram of what a starch looks like. It's not, we don't need to know like the exact composition of what a starch is. Just know that it's a very, very long string of sugars that are stitched together by specific types of bonds. And so specific enzymes can break those bonds. And the two that, or what, it's actually three enzymes that we're going to look at are alpha amylase, beta amylase, and 1,6-glucosidase. So there's three exoenzymes that a bacteria can secrete into the external environment, and between these three enzymes it can take this huge massive starch molecule and turn it into I think it's glucose and maltose. So the recipe is very straightforward, and all of these, it's very, we really only have to pay attention to what the substrate and indicator are. Everything else is just kind of a supportive addition, you know, like there's some nutrients. I think this one has like beef extract or something in it, and some agar to make the media semi-solid. So if we look at this, the recipe, the substrate is going to be starch, because if we're trying to see if it can eat starch, we have to include starch in the recipe. And then this one has an indicator that we're going to add after the fact. And the indicator actually is iodine. If you remember, if you took 107 here, we do a lab with iodine and starch. And when iodine and starch combine, I think it's called a Lugol reaction, or something like that. But when they interact with one another, they form this really dark amber color. So if we see the dark color, we know that starch is present. And if we don't see that dark color, we know that starch is absent and has therefore been broken down. So we can see a very clear, here's a typical plate that we would get. We see a bacteria up here that has no halo around it. Everything's dark. So we know that starch is still present, and therefore this bacteria would be negative. And here we can see a nice, very clear, this is a beautiful result. If only results were this clear in real life, it'd be great. But we see this beautiful halo around the bacteria, indicating that the starch has been broken down around this cell, and that this cell is most likely positive for those exoenzymes. So let's look at an example of some results. This is more typical of the results that we get, although we wouldn't, you're kind of wasting a plate by just doing one on a plate. You usually do, this is how we do it in lab. We typically do two per plate so that we can compare the results. So usually what we do is we have a negative control on the top or on the left or whatever, and we have a positive control on the other side, and then we put our unknown bacteria in the middle so that we can kind of look at the results all on the same plate. So the starch test is actually one of the interesting tests, or is an exception, I should say, to the test, because it's one of the few tests where you can't read an early positive. If you think about all of the tests in the biochemical testing, if as soon as your results are positive, you can be like, well, there you go, it's positive, no problem. You never want to read an early negative because you want to give it time to change. But if it's a positive, then great, you can read it. But the problem with the starch test is, of course, in order to read it, you have to add that iodine, and iodine's going to kill. You can't. If it's negative, you can't just add it back in the incubator and continue the incubation time because you killed your bacteria. So you really want to wait the entire incubation period to analyze the bacterial samples when you're doing the starch test, because as soon as you add the iodine, you're basically going to throw these guys in the biohazard bag because they're dead. And in all of these plated tests, the 4 tests that we're doing today, you really don't need an uninoculated control like you do in the tubes, because all you have to do is refer to an area of the plate that's not inoculated, right? There's so much space on these plates that you can see what an uninoculated media would look like. So here's our example of the 2 bacteria that we would do in the lab. We have A would be Bacillus subtilis. You'll notice that this guy Bacillus subtilis is positive for a lot of these exoenzymes. So you see that nice halo around the cell. And then E. coli is your classic. A lot of the enterics are negative for these exoenzymes, like E. coli is here. Whoops, I just gave you the answer. That's okay. All right, let's move on to our DNase test. So just like carbohydrates, it's in the bacteria's benefit, or best interest, I should say, to be able to break down DNA into those monomers, into nucleotides, and import them into the cell. Mostly not as a nutrient source, but mostly so that they don't have to spend the energy to make them. Remember that there's some, one of those off branches of the metabolic pathways that we talked about very briefly back in Unit 2 had some precursors that could be used to make the nucleotides. And that takes a lot of energy. And so if a cell doesn't have to make them, it's a huge advantage for that cell. So if they are able to take DNA that's in the external environment, chop it up and bring it into the cell, that's a huge advantage for them. So we're really looking, like always, hold on a second, I'm getting an important call. I need to answer this. Hold on. Okay, sorry about that. All right, let's go on to the second test, which is the DNase test. Remember that all of these tests, what they're really doing is testing for the presence of these enzymes, because enzymes are the protein that makes all of this possible. So the enzyme here is called DNase, and it breaks down DNA usually into like little dinucleotides, so little 2-unit pieces of nucleotides. And there's a slightly different mechanism of this depending on what bacteria you're looking at, but it's not terribly important to know the difference and the subtlety of that mechanism. Just understand that there are slightly different enzymes in different proteins, but they basically do the same thing. They break large strings of polymeric DNA, so polymers of DNA, into short little nucleotides that they can then import into the bacteria. So the recipe again for this is we of course have to have our substrate, which is DNA, and then we need an indicator, and in this case the indicator is included in the recipe. We don't add it afterwards. And that indicator is a very pretty dye called methyl green. It has this beautiful aqua color to it. And methyl green in the presence of long stretches of DNA is green. It basically forms this complex with the DNA. The DNA is very negatively charged, and it has some interesting chemical qualities because of that. And it turns this dye, methyl green, into this pretty aqua color. So if there's no long strands of DNA and they get broken up, methyl green turns a clear color, and so all you see is the agar. So in the presence of DNase, we should see clear agar. And if that bacteria doesn't have DNase, it's going to continue to be green. So let's look at some results here. So here's a result. This is actually a nice result. Oops, sorry. This is a nice result because the two that we're looking at in class is this S. marcescens and the Entero. I think we look at Enterobacter aerogenes or something, one of the Enterobacter species. And so you can see that Serratia marcescens creates that clear halo around the bacteria. It happens to be red. The bacteria itself is pigmented, and so when you grow it onto media, it turns red. It has nothing to do with the test. It just happens to be that bacteria is red. So this test is very clearly positive. You can see that nice halo. It turns out that Staph aureus is also another bacteria that commonly tests positive for DNase activity, and most of the enterics are negative for DNase activity. In practice, like if we were doing this in a lab, we typically don't get this nice, really beautiful, clear result. And we have to hold it over like a white sheet of paper or hold it up to the light in order to see this halo, and it's a really subtle halo. So it's really important to have a positive and negative control when you're looking at your unknown, simply because the halo can be really subtle at times. And what tends to happen is if this is a fast-growing bacteria, it'll create that halo within, like, say 12 to 16 hours. And then in the next 8 hours to 24 hours, like say you don't look at it for 2 days, that methyl green will just kind of like diffuse back into the agar and kind of make this halo not as clear. So timing is kind of important in the DNase test, simply because it's so easy to get a false negative. All right, let's look at lipids. So can bacteria eat fat? It turns out that they can. There's a wide variety of lipids, and therefore there's a wide variety of lipases. So the enzyme that we're looking for is lipase. And so depending on the lipase, that will determine what kind of lipid it can digest or break apart. And in this recipe, we're basically going to use a very, very basic type of lipid called tributylene oil. And tributyrin oil is basically a very simple, it's just got a glycerol head, 3 fatty acid tails, and it's just your kind of basic, it's the most basic and simple natural sourced fat that we can use. And it can pretty much be broken down by any of the lipases. So this will give us, we don't run the, how would I say this? We don't run, by using this tributyrin oil, we don't run a risk of having a false negative, right? So if we were to use, so we could, for example, we could make this oil, we could make this media with lots of different oils. And in fact, if you go to some diagnostic labs, they do that. They will make lots and lots of different types of lipid media, and then change the lipid that is as a substrate to see which lipids the bacteria can digest and which ones they can't. And then they can characterize the bacteria that way. So we're just going to do a representative example and use our basic kind of garden variety oil, which is tributyrin. Tributyrin also has a really nice aspect to it where as soon as we turn it into, remember that oils, all lipids are hydrophobic. So our media, of course, is made of water. So if we add oil to water, it's not going to mix. And if we were just to kind of put it in there and then pour our plates, we would have a mess. We would have media solidify, and then we'd have oil basically floating on top. So this agar is kind of a tricky agar to make. You basically have to make it, let it cool down, like, so, because you have to heat the water to make the agar dissolve in the water, cool it down to a certain temperature, add your tributyrin oil, and then you actually have to put it in a blender and emulsify it, kind of like, think about like shaking up salad dressing, right? If you shake it really, really hard, you're going to turn all those little oil, all the oil into tiny little droplets and emulsify it. And that's what we want, because when we emulsify tributyrin oil, it turns this really nice opaque color, which we can then use as an indicator. So the lipid test and the next one, the casein test, are kind of unique again. I guess it's not that interesting, but they're unique in that the substrate is also the indicator. So there's no special indicator that we have to use. We're simply looking for the presence or absence of, in this case, tributyrin oil. And I'll show you what I mean here in the next slide. So here you can see that the tributyrin oil is emulsified in the media and it's cloudy. And so when this tributyrin oil is used up, the media is going to turn clear. I shouldn't say used up, but broken down. The media will turn clear. So you can see a very clear positive result here, which is Bacillus subtilis on the right. And then we have E. coli and enteric here on the left, which is lipase negative. One of the things to keep in mind with lipid hydrolysis is that it tends to go bad really easily because it basically loses its emulsification, and the media will go from cloudy to clear in really sometimes just a few days, depending on the temperature that we're storing them at. So it's really important to include positive and negative controls to make sure that your media is working as it should. All right, our last test, proteins, only one left, casein hydrolysis. So casein is a type of protein, and we're looking for an exoenzyme called casease. And casease is going to break down proteins. It's basically going to dissolve those or break apart those peptide bonds that hold all the amino acids together. And casein is opaque. It's basically the protein that gives milk its cloudy white color. And when we break that down, it's going to turn our media clear. So the substrate, just like the lipase test, right? Our substrate is also our indicator. So the substrate is milk because, of course, milk contains casein. And our indicator is also milk because we're basically looking for the presence or absence of the casein. So here's some results. We can see a nice halo. Again, Bacillus subtilis has lots of these exoenzymes that it can secrete into the environment. So we see that nice halo where all the protein has been broken down around Bacillus subtilis, but E. coli lacks casease, and therefore we don't see that halo. All right, that does it. Oh, look at that. Under 20 minutes. Best one yet. I'm sure you're happy about that. Good luck on the quiz. And as always, email me or contact me if you have any questions. Take care.